that also has the highest complexity. Take a moment to review the table to decide the best system for you. Now we will review some tips for successful transitioning of existing feeder-dependent cell lines to feeder-free culture systems using the Essential 8 Adaptation Kit. The Essential 8 Adaptation Kit contains Essential 8 medium as well as a bullet of recombinant human laminin 521. In adaptation of proliferating feeder-dependent cultures, the cells are enzymatically passaged, they're harvested and washed, there's an additional wash step, and finally, cells are seeded into the feeder-free system on the matrix of interest. Here we show OCT4 whole well imaging staining 5 days post that transition into Essential 8 medium on various substrates. What you quickly observe is that the laminin 521 condition has optimum post-passage cell survival. This is further highlighted by the plot shown here, with the anti-site % confluency over time indicated. What you can see is that the laminin 521 shows the most robust performance in transitioning the cells from a feeder-dependent to feeder-free context. Finally, we'll provide you some information on the GEDC PSC cryopreservation kit in order to enhance your cell line security. The PSC cryopreservation kit comes with 2 components. It has a PSC cryo medium for cryopreservation of your cells and is also sold with a Revited Cell supplement for recovery of your cells to optimize the post-cell recovery. The PSC cryo medium is a ready-to-use formulation which is xeno-free, whereas the Revited Cell supplement that was covered previously, again, is an animal origin-free solution and is chemically defined. Here we show some data of cryopreservation of PSR-based cultures in the PSC cryopreservation kit, as well as using a homebrew method of using medium A plus medium B. What you can see is that in KSR medium alone recovery instances, you see nice recovery of the cells 6 days post-DOS. However, when Revited Cell is included for the first 24 hours post-DOS, you see a significant boost in recovery for both cryopreserved samples. Here we see the cryopreservation of feeder-free cultures using the PSC cryopreservation kit. What we can see on the leftmost panel is that we're looking at the 24-hour post-thaw viability for each of the solutions. The serum-containing solution using FBS with 10% DMSO is shown first, whereas serum-free Thermo Fisher Scientific solutions are shown next, followed by serum-free competitive solutions. What you can see is that the PSC cryopreservation kit, which is shown in the blue circle, provides optimum post-thaw viability. This allows you to have quicker time to experiment, as indicated by the plot shown below, with a percent confluency reaching passaging confluency much quicker in the PSC cryopreservation instance versus the best-in-class coupled with the 10 micromolar Y27632. The PSC cryopreservation kit is also very versatile for cryopreservation of your PSCs, allowing you to cryopreserve them either as single cells or using clump passaging methods. So we present a table comparing the PSC cryopreservation systems. As mentioned in this presentation, the PSC cryopreservation kit comes as a ready-to-use formulation that contains a recovery component, is xeno-free, it is manufactured also in a CGMP-compliant facility. Currently, we do not have a cell therapy systems version available. We do have a cell therapy systems available for this. With the information that's been provided to you in this deck, we've covered the pluripotent stem cell media systems available through Thermo Fisher Scientific. Using these systems, you're able to maintain your cells in reaching culture and maintain normal PSC properties, including morphology and pluripotency with SSEA4, OCT4 expression, normal karyotypes, as well as maintain the trilineage differentiation potentials of your pluripotent stem cells for your downstream differentiation experiments. If you're looking for additional opportunities through GEDC, please join us for our hands-on training courses offered at one of our 3 sites in either Carlsbad, California, Glasgow, Scotland, or Frederick, Maryland throughout next year. This course offers a 3-day hands-on training course, which is covering lecture as well as hands-on instructional information. It's limited to a maximum of 8 attendees per course for very personalized attention. Alternatively, you can join us at one of our virtual training labs, which cover pluripotent stem cell culture. You can learn it in a virtual environment and attend from anywhere. We have opportunities coming up at the end of this year and early next year. For more information, visit us at www.thermofisher.com/PSC workshop. Thank you for joining us for this session of 24 Hours of Stem Cell. Explore innovations in stem cell research with our second one tool. Experience next-gen virtual conferencing. Join fellow researchers for the third annual 24 Hours of Stem Cell event. Gain unprecedented access to prominent researchers and thought leaders worldwide. Connect directly with presenters to live Q&A sessions and panel discussions. Or take a conference to your schedule with on-demand presentations. Learn about the newest technology, products and resources developed to advance your area of interest, and save documents, presentations or resources to your personal database and review them at your convenience. All this from the comfort of anywhere. The field of stem cell research is ever expanding. Run with it. Embrace the technology. Collaborate globally. Confluence virtually. Today the world evolves around stem cells. 24 hours of stem cells. Register today to reserve your virtual seat.
Good morning and good afternoon, everyone. This is David Piper from Thermo Fisher Scientific. I'd like to welcome you to our keynote talk on genome editing and stem cell engineering for disease modeling. I'd like to thank Dr. John Chestnut for joining us today and to give you a brief introduction of his background. He received his PhD in cell and molecular biology from the University of California, Davis, and followed that with postdoctoral studies at the University of Colorado, looking at recombinant antibody technology. John's had really a broad experience in synthetic biology, I think before it was called that, really. When he started his tenure here at Invitrogen, he really was responsible for developing and productizing the topo and gateway cloning technology that many of you are familiar with, and that was a way to shuttle genetic content amongst many different vectors that were suitable for expression in a whole host of organisms. And he went from there to sort of the next logical step of defining targeting strategies using integrases to put material into pre-selected cassettes in various types of cells, but specifically and perhaps his main interest within stem cells. And that really was a jumping off point for him to begin leading the synthetic biology efforts and genome engineering efforts at Thermo Fisher Scientific, where he's now responsible for developing an extremely wide variety of tools that allow for the editing of cells, the detection of those edits, and downstream cellular analysis of them. And I think he'll give us a broad perspective of that today. I'd just like to follow up with a couple quick comments before he gets started. One, during the talk, please feel free to submit questions directly to John using the Q&A box located in the bottom left corner of your screen. And again, I'd like to thank you all for joining and invite you to continue your conversation around gene editing following the talk in our stem cell research booth scheduled chat in the exhibit hall.
Thanks, David. Welcome, everyone. It's a pleasure for me to give this keynote, this broad overview of a very exciting field. Today I'm going to just take a high-level view of some of the technology drivers that have really gotten us to this, I'll call it a tipping point of technology that really enables us to do some very exciting things to create new disease models to push forward that biological research and drug development. So I'll start with our first slide and really kind of take a broad view of genome editing. Basically, we have our chromosomal DNA, and we design vehicles that will direct certain activities to the chromosome, hopefully specifically and efficiently. The activities, these enzymatic activities or fluorescent activities or labeling activities, could be activators, transcriptional activators, they could be transcriptional repressors, they could be labels, as I mentioned. Today what we're going to discuss is specifically delivering nucleases that create breaks in the DNA. So the important part is that these tools will create, in our discussions, will only create double-stranded breaks. And the more efficient and specific we are creating these double-stranded breaks, only at the locus that we're interested in, the better off the workflow is downstream. So once we create this double-stranded break in these cells, we can rely on the cell's repair machinery to repair the break. And the mammalian cells tend to be somewhat sloppy about this. So then what generally happens at these repair sites is that there's an insertion or more often a deletion of genetic material at these sites during the repair process. If we create this double-stranded break in a protein coding open reading frame, then this sloppy repair will more often than not result in a frame shift in the reading frame, leading to a premature stop codon and effectively knocking out that gene. If, along with our nuclease, we deliver DNA that has homology to either side of the break, we can replace DNA, so we can either repair single nucleotide polymorphisms, mutations, or we can induce model mutations into the cell line. If we use some larger donor DNA that has homology to both sides of that double-stranded break, now we can add either new elements, including labels, or new genetic coding regions. So really, the crux of the entire genome editing issue or problem is creating this double-stranded break in the genome efficiently and specifically. Application-wise, there's a broad amount of applications that we and others are interested in. Today, we'll focus on cellular disease models. We can create the disease models in animals as well. Cells at factories, so we can modify CHO cells or other cells that are creating biopharmaceuticals to make more robust drugs. As we get better at drawing out diseases or tissue models in 3 dimensions, we will use what we learned in the cellular models to create the tissue disease models. And then also, there's a lot of work going on in transgenic plants to create disease-resistant and more robust crops, as well as carbon sources for biofuels. And then, as we learn along these other applications, will eventually be, as a field, pushing into gene and cell therapy. And I just have one parting shot on that. But generally, we can study gene function. We can target gene mutations, as I mentioned. We can add transgenes into the genomes of animal models. We can label, we can add stable integration, and eventually tissue and cell engineering for novel functions. So if we look at the cell engineering cycle, as we call it, as a synthetic biology group, we look at it as type of a design, build, and test model. So first, if we have our cells, we want to understand the issues that confront us or what the type of experiments we want to do. One of the main drivers I'll discuss is sequencing. So next-gen sequencing is providing volumes and volumes of data as to what the genetic makeup of a particular cell line might be. And now we can use that data to be able to understand the types of mutation profiles that particular disease models contain. We also can use our in silico tools to take this data and then help us design specific tools that we're going to use to create our edited genomes. Once we understand how to create our tools, we can then use DNA synthesis to create some of the pieces that we want to put in, the donor DNAs. Eventually, as the field matures, we'll be able to put in larger pieces of DNA, including, I think, genomic circuits that we'll be able to assemble from a parts list of genomic parts. Then getting down into the genome editing tools is another of the technology drivers. As you probably know, we will hope to convince you later on in the talk that these tools are becoming much more